Hi, welcome back to the Sound of Science, AP Edition. Today we're going to be talking about cellular transport, how materials pass from one side of the cellular membrane to the other, either moving into the cell or out. As a quick review, the plasma membrane is made up of phospholipids that are amphipathic. Essentially, that just means that they are hydrophobic on one end and hydrophilic on the other. As a result, they're going to form this bilayer whenever they're in an aqueous environment. They do this spontaneously. And because of the nature of the phospholipids, they're actually able to move. It's a fluid model. It is not that they line up and they stay just like that. They actually are moving constantly, which really gives cell membranes a lot of freedom. The membrane itself is what we call selectively permeable or semi-permeable. Basically, it means some things can move through the membrane, some things can't. Small and non-polar molecules typically are going to have no problems moving through the membrane. They just squeeze through those phospholipids, no problems. Hydrophilic substances, large polar molecules, ions, they can't move through the membrane that way. Primarily because of the nature of those phospholipids. So they're going to use channel proteins. Think of them like slides. They're just a tunnel. Hydrophilic tunnels, they go all the way through the membrane. Specific target molecules are able to pass through them. Or we might have a carrier protein. Carrier protein also goes all the way through the membrane, but this one changes shape to move a target molecule from one side to another. Switch it up a little bit. Let's move to cell walls. Let's talk just a little bit about the cell wall, which is found in plants. It can also be found in fungi. It's also found in bacteria, which are prokaryotes. So how is the cell wall and the cell membrane different? Well, the cell wall is going to produce a structural boundary for plants and other organisms that have cell walls. It's going to help protect and maintain the shape of the cell, and it prevents the cell from rupturing when water pressure is high, such as turgor. Turgor gets too high. In addition, especially with things like plants and fungi, it's going to help it stay upright. It's actually providing structural support to the entire organism. But it's also a permeable barrier. It has these tiny pores, these small holes called plasmodesmata, and they occur between the plant cells, and it actually allows for waste and water and ions to move freely back and forth between cells. So just to give you an idea of the different types of cell walls, because not all cell walls are made the same. All cell walls are going to be made of complex carbohydrates, that we know. Cellulose is a polysaccharide. That's what we're going to find around plant cells. Fungi have a polysaccharide cell wall as well, but this one is chitin. It's a different polysaccharide. Prokaryotes, bacteria, they're actually going to have a cell wall produced with peptidoglycan. This is going to use sugar and amino acids to produce the cell wall. And of course, this is a little different if we are talking about archaea bacteria, which is another class, another whole domain of bacteria that lack peptidoglycan. Back to cellular transport across the membrane. When you have a selectively permeable membrane that separates two areas, and there is a solute that's got a higher concentration on one side than on the other, what you've created is called a concentration gradient. You have a high concentration of solute on one side, a low concentration on the other. And as a result, you're forming almost like a ramp, right? So molecules are going to naturally move from areas of high concentration to areas of low concentration, primarily because that is what will make the cell most comfortable. So with movement across the semi-permeable membrane, we actually have a couple of types of transport that can occur. And so to begin, we're going to start by splitting it into two categories: passive and active transport. Passive transport is what happens when molecules or solutes move from a high concentration to a low concentration without the use of energy. No ATP required. They just happens. Whether it's moving through the membrane itself or whether it's going through some type of channel, regardless, it doesn't require energy and we move with the concentration gradient that we created. This is primarily how materials are going to be imported and waste are going to be exported. Diffusion is the movement of these particles along the concentration gradient without energy. It's a type of active transport. Small, nonpolar molecules such as oxygen, nitrogen, and carbon dioxide are going to move this way into or out of the cell. In facilitated diffusion, we're still moving along the concentration gradient. We're still not using energy. But in this case, we're going to use transport proteins of some type, either some type of channel or just some type of protein that's going to help get through this membrane. And that's why we call it facilitated diffusion, because it requires help. This is what we're going to use for things that are hydrophilic molecules or things that are ions. So like I said, facilitated diffusion is going to use transport proteins. It's, again, going to be for the movement of polar molecules, charged ions, and actually large quantities of water. Small amounts of water can actually move through the membrane, but a large quantity of water is going to use a channel known as an aquaporin that allows water to sort of flow in a little bit more freely. Switch gears and let's talk about that other type of transport, active transport. The first type that I want to talk about is actually endocytosis. And this is just where part of the cell membrane creates a new vesicle around something that is in the extracellular fluid and pulls it in. So endocytosis is going to use energy. You have to have energy to move that amount of membrane. There's really three types here. The first is phagocytosis, which is just where it surrounds large particles. Think of macrophages in your immune system, where they surround whatever invader they're trying to consume or destroy. Or think of an amoeba surrounding its food source to eat it, creates that vesicle. Penocytosis is where it's taking in fluids, the extracellular fluid, usually with some sort of dissolved substance that the organism needs, that we call penocytosis. And then, of course, we're going to have a receptor-mediated endocytosis. So essentially, this is where you have some sort of receptor protein on the cell membrane. And when a target molecule attaches to that receptor, it stimulates the membrane to surround it and create a vesicle. And so that case, you're only taking in items that the cell particularly wants or particularly needs. All right, so let's switch gears and talk about the reverse of endocytosis. This is called exocytosis. And literally, this is when you have an internal vesicle. It's going to use energy to fuse with the plasma membrane and secrete large macromolecules out of the cell. So this could be things like proteins, signaling proteins, hormones, waste, those sorts of items. And as you can see in this diagram, this is what the vesicle looks like with its phospholipid bilayer. It literally becomes part of the cell membrane so that these items can be secreted, they can be released. Active transport with carrier proteins is a little different. So again, still active transport still requires ATP, but essentially it is proteins that change shape. And we refer to these proteins as a pump. Requires metabolic energy. And just in case you needed the reminder, ATP means adenosine triphosphate. We just abbreviate it because adenosine triphosphate is kind of a mouthful. But this type of pump helps establish and maintain a concentration gradient. And in addition, it's always going to be moving against the concentration gradient. There's another type of active transport known as co-transport. And co-transport is just the secondary type. It uses energy from an electrochemical gradient and usually is going to transport two different ions across the membrane again, with the use of a protein. That's why it's a part of active transport. So there's two types of co-transport, kind of. Symport and antiport. Now, but symport and antiport, in these cases, it's two different ions that are being transported. Symport, both ions are going in the same direction, either out of the cell to in of the cell to the inside of the cell, or from inside the cell to the outside of the cell. Anti-transport, they're actually moving in opposite directions. It's two-way flow. So the cell membrane, which allows for the formation of these gradients, can also become polarized by movement of ions across the membrane. So it creates this electrochemical gradient. It's a type of concentration gradient. We also might refer to it as membrane potential. There's actually an electrical potential difference. There's voltage across the membrane. You actually might think of this when you come to muscle cells and their contractions. This is actually all related to that. And this is also why we can use electricity to change the rhythm of the heart. And of course, you guys hopefully know at this point that you can't restart a heart that has stopped, but if the rhythm is wrong, you can actually fix that rhythm with an electrical charge. So I've got a diagram here, and I also have a video, and this video is about a minute long, and I'm going to include it with this because I feel like it gives you a better idea than the diagram. Okay, so in that video, it showed you how the sodium and potassium basically are exchanged using this pump. Well, that exchange is actually going to create that electrical potential that we see in the membrane. And that is all we had time for today. Check out the next video to learn a little bit about another type of transport called osmosis, which is the movement of water. Make sure that you understand all the concepts that we went over here. Make sure you understand how the cell membrane functions to allow transport into and out of the cell. And make sure you understand how the use of energy is required for some movements, but not for others. As always, I'll catch you next time on The Sound of Science.